Welcome to the 2024 Research Grant Program overview. So the purpose of this webinar is just to enhance your familiarity with this year's program and to talk about some of the details. So I'm Jill Lawson. I'm going to be doing this introduction. I'm the Research Grant Program Manager. And then we will move into each of the funding priority areas for an overview of each priority. And we'll complete that with some instructions on the application process. And then we'll finish up with a question and answer session. So just a general overview about the Good Food Institute. We're a 501c3 nonprofit, and we're working to build a roadmap for a sustainable, secure, and just protein supply. So that focuses us in 3 areas: science and technology, corporate engagement, and policy. And that is encompassed in 6 different regions: United States, Brazil, India, Europe, Asia Pacific, and Israel. And our competition is also a global competition. So we accept proposals from all over. So this is a slide about specifically about our research grant program. So we're interested in accelerating the development of alternative proteins and making products that are delicious, affordable, and accessible. So this year, our program specifically chose topics in cultivated meat, plant-based, and fermentation-derived proteins that would be able to address technical bottlenecks, unlock additional research funding, and would foster collaborations to expand the field. And then we have some information about our program to date. We've awarded over $12 million to 118 research projects across 21 countries. So we provide support for the whole ecosystem. So that means that we are looking for open access research, and we want that to be widely used in order to get the most impact. So we have intellectual property terms in our grant agreements. Those will differ for foreign nonprofit entities. I encourage you to use the link in the RFP to our standard grant agreement to become familiar with what those terms look like. And at the end of the RFP, in the frequently asked questions section, we provide a lay summary of our IP terms. We encourage information sharing, but we do not prohibit generating IP or protecting IP. The program this year is $3.5 million across our 3 topic areas. The closing is May 23rd, 4 p.m. Eastern Daylight Time. And our 3 priority topic areas are upcycled plant proteins, next generation fermentation downstream processing, and hydrolysates for cultivated meat. These projects should be 24 months or less, and the budget cap is $250,000. Additional funding is available, $50,000 for collaborations with researchers that are new to the field of alternative protein. So I'm going to introduce Pri, and she's going to give you a peek under the hood of our priority topic 8.
Amazing. Thank you so much, Jill, for laying that groundwork. I'm so excited to talk to you guys about project topic A of upcycled plant proteins. Next slide, please. Okay, so before we dive into the specifics of the topic, here are some broader impacts just to kind of set the scene for applying upcycled side streams for plant protein production. So generally, I think we all understand that food waste and food loss are a huge global crisis, especially considering how many people go hungry across the globe. And when we're specifically looking at food manufacturing, it is estimated that approximately 39% of food is lost. And this is just during the food production process. Alternative proteins have so many opportunities with synergies of other sustainable food and agriculture practices. And this proposal focuses on one of those synergies. Can we reduce food loss and increase value of the whole crop by using side stream byproducts to form valuable food ingredients? On the right here, we see a few examples of volumes of protein-rich processing residues produced in very large volume. This is specifically for North America. Obviously, different parts of the globe will have different crops that are being generated at these high volumes. But we do see that there's millions of metric tons of these byproducts being produced annually, and we really have the opportunity to make them high value while creating novel plant-based foods. If the right byproducts and processes are used for these plant-based foods, this could potentially lead to making the whole process more sustainable, improving the price, and improving the scalability of plant-based foods. So for this proposal, side stream optimization will focus on all three of the following. So firstly, identifying a promising side stream from a food manufacturing process. So it has to be a food manufacturing process that has preliminary evidence as a viable alternative protein ingredient. Secondly, it must have optimizing raw material processing using some kind of emerging method. So how can we actually get this side stream into a food ingredient with new processes that are even better than the traditional alkaline extractions, acid precipitations, alcohol precipitations, et cetera. And finally, developing volume and economic models for optimized processes. Note that while we believe there are definitely many, many ways to get valuable side streams and upcycle them, here we are focusing really on side streams from food manufacturing and not outside industries, mostly because we're trying to set these up for quick commercial success. And we want to make sure that these ingredients are food grade. So this proposal is a little different than traditional explorations of side streams because we want to ensure that the projects are set up for strong commercial success. As a result, we are requiring evaluations of potential volumes of the side stream being explored, as well as economic evaluations. These do not need to necessarily be full forecasted production volume models or techno-economic assessments, although more sophisticated models will likely score higher. We are mostly looking for a strong understanding from the researcher of the potential production volumes, showing that the volumes are indeed high enough to be feasible to contribute as a food ingredient. And the economic evaluation is meant to display potential economic bottlenecks or areas of improvements for the side stream processing for researchers to take on after this project is complete, or even while the project is being completed. So I highly, highly recommend reading the recent side stream analysis that GFI released. I'll put it in the chat once I'm done speaking, and it's also on the last slide that I'll be talking about here in a bit. But I highly recommend taking a look at that side stream analysis because it'll really give a good gauge of what type of volume and economic evaluations are appropriate for this proposal. And note that we are not necessarily requiring an end product prototype of a full plant-based food from the side stream ingredient. But that being said, definitely showing some kind of functionality of the side stream to show that it could be part of a plant-based food is a must. Oh, thank you, Lucas, for doing that. So Lucas just put in the side stream report and highly, highly recommend if you're applying for this research priority, please read that. And so to reiterate, what these must include, the proposals, which side stream raw material and emerging processing techniques are planning to be used, what preliminary research has been conducted on this raw material and processing techniques, either inside of your lab or outside of your lab, how they will conduct a production volume calculation of the side stream raw material, how they will conduct an economic assessment of their process, highlighting bottlenecks that could be improved. And then optionally, but encouraged, how will they apply this side stream ingredient to an end product prototype? And importantly, slides that we will not consider, or proposals that we will not consider. So please be mindful of this. Solutions that attempt to use side streams from non-food industries or non-food safe ingredients, reagents, or processing technology. Again, sorry to limit the scope. I know that there's a lot of non-food industries that have excellent byproducts that we could be using in our industry as well. But just to smooth the transition for these side streams as much as possible, here we are focusing on food industries. Additionally, we won't consider solutions that attempt to use food side streams without a clear vision of how it would be produced into a plant-based ingredient. I know it can be really difficult working with a novel raw material to kind of pigeonhole it into a certain application, like a plant-based food ingredient. But in this case, we really want that preliminary evidence that shows that it really could be used as a plant-based food ingredient. Additionally, we won't consider solutions that attempt to use food side streams being currently produced at really negligible volumes or without commercial potential. So please stress how your side stream has commercial potential in your proposal. And then finally, solutions that rely on AI approaches without rigorous experimental validation. And finally, where can you learn more? There's a lot of resources that you can dive into on side streams that GFI has identified, but we'll definitely look at side streams that we haven't identified. We're really open to that. We just need to be a little convinced in the proposal that it's going the right direction. And again, I really highly recommend reading this side stream analysis that Lucas linked in the chat, and that's shown here on this slide. Because this topic priority area was inspired by this work in particular. So you can imagine a lot of the work and recommendations would be really well aligned with this proposal. And that's it. I hope you guys apply, and I'm really excited to see what you come up with. And with that, I'll pass it on to Lucas.
Hey everyone, thanks Pri. So this year I'd like to introduce our topic for fermentation this year, which is focused on downstream processing and specifically precision fermentation protein isolation. So as many of us know, the goal of upstream bioprocess is to maximize the product yield, which then allows us to improve DSP efficiency and overall costs. But while increasing the amount of product generated in the upstream process through strain development, feedstock improvement, process improvement can improve COGS, there's also significant opportunity for cost reduction in downstream processing as well, or DSP. So the goal of any fermentation downstream process is to really recover, concentrate, and purify, then finish, formulate, dry, and store your food ingredient of interest, whether it may be a biomass or a precision fermentation protein product. So the process tree on the right really shows several downstream process possibilities depending on the product and the localization, whether it's biomass, intracellular, or an extracellular protein. For biomass products, this is just a couple of steps. Usually you have a harvest, maybe a heat treatment for RNA reduction, then a drying and a storing of your product. However, for precision fermentation proteins, the protein has to be recovered from either the broth, so the extracellular secretion, extracellular matrix, or the cells, so intracellular localization of that protein ingredient. So intracellular production can increase the processing steps due to the need to disrupt and extract the protein of interest. So how do we isolate proteins in precision fermentation? Well, you can do it through solubility, such as isoelectric precipitation, size and charge, like typical membrane filtration, TFF, or by charge and binding specificity, such as ion exchange and adsorption chromatography. And each of these really has a capital cost, an energy cost, and a water usage cost associated with them that can really impact the technoeconomics as well as the environmental sustainability of your process. So really current precision fermentation DSP is a multi-stage unit operation system to separate the biomass, extract proteins, concentrate, purify, and dry the material. And so the increased number of unit operations really increases your capital costs, your energy consumption, your water consumption, and really your loss between stages, which all impact the downstream yield and unit economics. So if you can imagine a DSP process that has 4 unit operations, so 4 separate unit ops, whether it's on separate equipment or separate stages, each step has its own step yield. So let's say 90 to 95% is your yield. So that means you've recovered 90% of the biomass or the protein present from the prior step. However, subsequent unit steps will sequentially reduce your total protein yield. And a process with 4 unit ops with like a 95% step yield will only result in an 80% overall yield from start to finish. So maybe each step drops to only a 90% yield. Now your total yield drops to only 65% with 4 unit ops. But maybe you need to add an extra filtration, a polish, or a wash, and maybe that 65% yield now drops to 60% yield. So this is valuable product that is getting lost over time during a DSP operation. So really, as you increase the number of unit operations, you lose product and you increase costs due to labor, electricity, water, etc., as well as capital and equipment. So we're losing that product, which impacts your cost of goods sold, and not to mention the environmental impact because you're using more energy and water. So it's really no surprise that DSP can account for a significant portion of your operating costs, as well as the capital costs, depending on the amount of equipment are needed. So we wanted to focus this year on specifically precision fermentation proteins and DSP operation improvement. So a proposed solution for this would be to reduce the costs and increase the sustainability of precision fermentation derived DSP, would be to develop novel approaches for protein isolation and drying that improve these unit economics. So ideal solutions could be integrated one-step capture and purification of targets. So this would be like a high affinity and selectivity, but reducing the cost since chromatography has high water and high capital costs. It could be high throughput or a semi-continuous processing that has a lower energy consumption. These continuous or semi-continuous DSP solutions could be another enabling area to complement continuous upstream processing as well. So this all needs to help with DSP scaling with size. So solutions could be novel single capture step affinity. They could be filtration equipment architecture improvement. They could be novel separations, substrates, or materials. They could be biochemistry solutions that improve the separation and isolation, such as different precipitation mechanisms or a combination thereof. But we are focusing on precision fermentation proteins specifically for food ingredients and alternative meat products. So when we're looking at proposals this year, we're looking for several things to be articulated. Really, the solution must be for fermentation-derived alt proteins that are specifically for precision fermentation for food. And we're even looking for a tighter focus on precision fermentation protein ingredients for alternative meat products that improve the organoleptic properties of our plant-based meat alternatives or fermentation-derived ingredients. So these process improvements could be optimized, adapted, or novel unit operations for DSP in the form of equipment or consumables, such as filtration architecture or flow improvements, pump improvements. They could be materials. They could be membranes or filter substrates, affinity-based systems, affinity molecules, or a hybrid. That could be a biological, a chemical, or an engineering solution that combines both equipment and processes. Example could be isoelectric precipitation. So we're also looking for the proposed solution to have demonstration, development, or engineering of the technology as it specifically applies to food protein DSP for fermentation-derived alternative meat proteins. So this can be for really any precision fermentation protein that enhances the organoleptic properties of meat alternatives, such as flavor, color, or structure enhancing proteins. Just as an example, collagen or heme proteins, but not limited to those specifically. And any demonstrations should be with these relevant food proteins. We would like to see how your approach will significantly improve the unit economics over current DSP systems. Maybe this would be an electrical model or a water consumption model per kilogram of isolated protein. And maybe you could project that to larger scales. We'd also like to see a path to scaling the solution to commercial scale bioprocess unit operations, just so that you can acknowledge the risks of scaling and mitigate strategies in the proposal. So a few things that we will not consider. The first is AI or machine learning approaches or screens without substantial hypothesis-driven frameworks or underlying training data to produce physical and tangible deliverables. So solutions focused on higher value ingredients like growth factors or enzymes or other food processing agents will not be considered. We're, again, really looking for ingredients that are geared towards alternative meat products that can improve the organoleptic properties of our meat alternatives. So in this case, we are not looking for precision fermentation caseins or precision fermentation egg proteins. If your technology can be applied to those classes of proteins, that would be great. We want to see proposals that can be effective for multiple classes of proteins, but we would love to see demonstrations that are for meat alternative precision fermentation proteins. So other things that will not be considered are solutions that don't necessarily improve the water or electrical consumption. Maybe it does improve the process yields and times, but if you can show improvements to electrical and water, that would be great, or maybe even reduction in capital. Solutions that do not implement food safe materials will not be considered. We're really looking for applications that can be scaled to food grade GMP systems. And then other areas of technologies that lack a pathway to commercial scale. Again, we're seeding early stage research, but we want you to show a pathway to scale for that technology, or at least the steps that could be taken to reduce cost of those technologies. And then finally, we're looking for technologies that are effective for multiple classes of proteins. I know this is very difficult to do at times, depending on the process, but if you can show applications to other protein classes, that would be terrific. Or if it's only geared towards one specific protein class, we might have to not keep it in scope for the proposal. So please apply your proposals to alternative meat precision fermentation protein ingredients that improve the organoleptic properties of our meat alternatives. I'll just leave this up here. There's a few resources. You can check out our fermentation capacity analysis. In addition to just a couple of resources that we put in the RFP, we can throw these additional examples into the RFP so that you all have those. Those show several different examples. Please don't be limited to these specific examples, but this would give you a sense of different types of technologies. And with that, I'll hand it off to Elliot for topic C.
All right, thanks, Lucas. So topic C is about hydrolysates for cultivated meat. So the impetus behind this particular research priority is that based on the modeling efforts that have been done thus far in the cultivated meat field, they suggest that amino acids are predicted to be a cost driver. So in particular, amino acids, when you're growing proliferating cells, a lot of that biomass comes directly from these amino acids being consumed by those cells. And today, amino acids come primarily from individual fermentation processes. And so if we were to source amino acids for cultivated meat at scale using these individually fermented amino acids, they would be a major cost driver in the production of cultivated meat. And furthermore, basically, in order for those costs to come down, there would need to be demand on the other side. And presumably a larger production volume of cultivated meat would need to be the reason for that demand. And so there's also a catch-22 that we're left with, which is that amino acid costs really only become low when the cultivated meat is large, but the high costs during the early stages of the industry could likely prevent the overall growth. So on the next slide. It's been proposed that amino acids could potentially come from hydrolysates to alleviate, at least, partially mitigate this bottleneck. Hydrolysates from different raw materials like plants or animal sources or microbial sources have been used as supplements in cell culture for decades. However, the hypothesis with cultivated meat is really that we probably need to have hydrolysates be the primary source of amino acids. And so what does this actually mean in practice? You can see an example here to the right of this slide, where if you look at the essential amino acid profile of a soybean meal, which is shown in orange, compared to the amino acid demands of a hypothetical cell, you can see that soybean actually has a fairly favorable amino acid profile that would meet the essential amino acid requirements of the average cell, although it's deficient in certain amino acids. And so if we were able to actually use hydrolysate, so in this case, soy hydrolysate, for instance, as a source of amino acids at a cost of around $2 per kilogram, which was modeled in this one particular study, then you can see that the cost effect that it would have on this macronutrient category, which is primarily the basal media components in cultivated meat, would be quite dramatic. And furthermore, it could also result in fewer raw materials being needed overall for media formulation that could result in a more simplified supply chain with fewer intermediate players. So what is a hydrolysate? It's essentially taking a proteinaceous raw material and then breaking it down into its constituent components through a process known as hydrolysis. And this process can begin with heat, use enzymes, as well as adjustments in pH to break down these proteins into free amino acids, individual peptides, as well as other components that get carried over in the process, such as minerals, carbohydrates, lipids, etc. So essentially what you're left with throughout this process is a product that is a supplement or a nutritional feedstock for cells. And basically what we're looking for in this priority area, next slide, is proposals that can help us advance hydrolysates for use in cultivated meat in general. And so proposed advances or proposals that we're looking for, they could actually fall along various different processing stages from the diagram that I just showed you. So you might look at different raw materials being used, whether those are microbes or algae or other commodity crops or side streams, as Pri was talking about before. You might want to figure out what is the best method of hydrolysis, the best way to apply heat, filtration, and purification to result in an end product that is suitable for cultivated meat. And so I've listed here some examples of open questions that I would consider attractive to interrogate for this priority area. So as I mentioned before, what raw materials should we actually be using? How do those fit the nutritional needs of the cell? How do they perform in cell culture? And are they suitable for cultivated meat in terms of cost, sustainability, and consistency? You might think about optimizing the actual protocol parameters to create the hydrolysate. So what enzymes might you use? What protocol parameters might you apply or change to achieve a desirable degree of hydrolysis and nutrient availability? It's been known that, depending on your raw material source, you might have anti-nutritional compounds or growth inhibitory peptides that are sort of residuals that get carried over that can cause bad batches in your cell culture. So how do we identify those? How do we minimize those? How do we mitigate that? How do we limit batch-to-batch variability, which is also a challenge for certain hydrolysate production? How can we lower the QA and QC burden to ensure that we're getting consistent batches over time without overburden of analysis? And what methods are most fit for purpose for cultivated meat, and what can actually be scaled for production with acceptable cost profiles? So we're looking for proposals that really successfully articulate what raw materials you're planning on using for testing. Again, these have to be non-animal source materials. They can be any non-animal source materials, but they can't come from animals, no matter if it's like an eggshell or a bone or whatever. It has to be non-animal. Please include, what cell type, what species you might be testing on. You might want to tailor the raw material to your specific metabolic requirements of those cells or those species based on experimental data or data that might be available in the literature. You should have a clear hypothesis of how your proposal will enable lower cost and higher performing hydrolysate ingredients, again, that are suitable for commercial scale production. We also want to see, the experimental approach clearly outlined for testing these hydrolysates in cells that are relevant to cultivated meat. If you're unclear about what cells you might want to test on, there is a link there to a report on cell lines where we surveyed the industry that sort of overviews what cells are most attractive for companies, what are maybe most, what are most attractive from a commercialization standpoint. Any controls or comparators that will be used. So again, we want to make progress, hydrolysates already exist, but we want to make progress in their application in cultivated meat. So it's important to have, controls and comparators that you're going to be using. Discussion of limitations or uncertainties in the devised approach. Maybe you're proposing something that hasn't necessarily been done before, or maybe it's a different, taking something from one field and applying it here. So please discuss, some of that uncertainty that may exist, what you might do to mitigate that uncertainty, etc. And finally, consideration of how costs, energy use, or other resource utilization might otherwise be affected or could be improved compared to, again, existing practices for hydrolysate production. So finally, proposals would be considered out of scope if they, first of all, we're looking for at least one primary aim to improve the processing or characterization of hydrolysates. So if you're simply trying to test, buy a bunch of hydrolysates off the shelf from some manufacturer and screen them in muscle cells from like a cow, that's going to be out of scope for this particular priority area. We really want to have, at least a portion of the aims of these proposals to be focused on improving the processing or characterization of these hydrolysates. So certainly you can screen existing off the shelf hydrolysates performance, especially if you're doing comparative studies, but they cannot be the sole focus of the proposal. As I mentioned before, any animal-derived raw materials cannot be used as a source for hydrolysates. You may use animal serum in media formulations, specifically if you're using cells that might not have open source serum-free formulations, especially in the cultivated seafood arena. Or in particular, if you're looking to do comparative studies to understand how the hydrolysate might assist in advancing the development of serum-free media. And then finally, this probably should go without saying, but don't use any materials or processing inputs or aids that might be expected to cause food safety or other regulatory issues. Turn it back. Final slide is just some more information. I've listed some studies with, I think, relevant literature that's, definitely would it be informative for reading before applying to this particular priority area, as well as the side streams analysis, which has somewhat of a breakdown of how to apply side streams for hydrolysates for cultivated meat. So I'll turn that over to Jill.
So I'm going to go through some of the application process with you, and hopefully that's going to answer some of the questions that are outstanding. So in order to start a proposal, there's a link here, but also the easiest place to get this is the cover page of our RFP. So if you go to our website and you look at our RFP, the very first page has a link. When you go to that link, it's going to ask you to set up a user profile before you can start a proposal. There's instructions there. If you have submitted with us before and don't remember your password, there's instructions on how to manage that right there at that link. What we're going to ask you for are some basic project details, like information about your organization, your title, a summary of what you're proposing, key personnel, information about human subjects, animals, hazardous material. Then we're going to ask you to upload a research design, which is a 6-page limit. One of those pages will be describing your research team, summarizing qualifications and background. This is in lieu of uploading CVs or including bio sketches. So you're going to condense that down and describe the important aspects of the people that you are working with. And then up to 5 pages is going to be describing your project aims, your research design, your methods, collaborations if you've got them, and that should be inclusive of your figures and references. So work plan, will be a one-page upload that is going to outline your project objectives, milestones, tasks, dependencies. Generally, people are a fan of using like a timeline or a Gantt chart. There's no specific defined method that needs to be used. Budget is one page. There is within the application instructions, within the application itself, there's a link to a budget template. You'll open that, you'll save a copy, you'll complete it. One page. You're going to itemize your costs within that budget. You'll include one page per subaward. So if an additional page is required for every organization that will be receiving a subaward from you. So if you have a collaboration with another organization to do part of this work, then they will have a one-page budget that is included in your in your upload. So you're going to itemize your costs. Costs are going to be in US dollars. Don't exceed $250,000, $300,000 if you have a qualifying collaboration. And again, those should be in US dollars, and indirect costs should be included within your total budget amount. And just FYI, that is a maximum of 10% is what we allow for indirect costs. Budget justification is 2 pages where you're going to detail the items that you have included within your budget, including subawards. If you're requesting that additional $50,000, this is a great place to detail how your collaborator is qualifying as new to the field, which is basically our definition of new to the field is not having received any type of funding for work in the field of alternative proteins. So base your budget on actual estimated costs and request the funding required to be successful in completing your project, but be conservative and realistic with this budget amount. Your impact strategy is a 2-page limit. You're going to provide a detailed description of your plans to achieve meaningful outcomes and maximize the impact of the proposed research. If those plans include something that incurs a cost, those costs can be included in your budget. And I encourage you to look at the frequently asked questions section at the end of the RFP. We have some helpful prompts if you are wondering what should be included in an impact strategy. And letters of commitments are optional. Each letter could be 2 pages or less and should be combined into a single document. You would include them if you have a collaborator that's committing financial or in-kind resources. The letter should describe their role and the estimated monetary value. You may also include a letter of commitment if your organization is committing resources or in-kind contributions to your proposed research. And you would also include a letter if the principal investigator is a graduate student or a postdoctoral fellow, and that letter should be from a faculty member that is committing to act as an advisor throughout the term of the proposal. And it should confirm that you have the laboratory facilities to complete the project. And for student-led projects, it should also describe the aims of the student's dissertation and how this proposed research contributes to them. Proposal submissions are due May 23rd. by 4 p.m. Eastern Daylight Time. No proposals will be accepted after the deadline. Proposal uploads have to adhere to the stated page limits. Proposals that don't follow the guidelines will be excluded from the review process. And then proposals that have been submitted through our proposal portal are still accessible. So if you submitted a proposal, you may still log in and make changes to that proposal up until the deadline. So you don't need to contact us to make any changes. This is a slide discussing our review process. So all proposals that are submitted move through a comprehensive evaluation process. It starts with the compliance review, then it moves on to a pre-review proposal screening. If the proposals don't adhere to the guidelines, they don't meet the topic inclusion criteria or are significantly scientifically or methodologically unsound, they won't move on to the next phases. The next phases are external peer review assignment, a global GFI review panel, and finally a funding committee to make final funding decisions. I want to point out that we are accepting global applications, and that means that our requirement that proposals be submitted in English means some people are going to be non-native language submitters. And I just want to provide an assurance that proposals are evaluated on scientific merit and not language proficiency. So the evaluation criteria are as follows. Originality and significance are the primary review criteria. So you can see that's novelty, innovation, and transformative potential. That's relevance to the funding priority area and suitability of the proposed methodology. The rest of these are all secondary review criteria. So we'll be looking at your research team, the team's ability to deliver the proposed project, expertise and background of the researchers, balance of skills within the team, and we'll be looking for strong, meaningful, and synergistic collaborations. Project planning, we're going to be looking for clear, well-defined research objectives, proposal methods that are clear, feasible, and capable of producing reliable results, and well-structured plan detailing the research stages, timeline, milestones. Value for money. We're going to be looking for reasonable budgets and transparent justifications. Research value for the budget requested also. And expected impact. How comprehensive, feasible, and creative is your impact strategy? And what is the potential for achieving real, meaningful, and broad impact? So I encourage you to check out at the end of our RFP is a frequently asked questions section, and that section of the RFP, the frequently asked questions, will be updated with commonly asked questions. So if you've read it once, you might check back and see if any of your current questions are listed in the frequently asked questions. You can send questions to this email address. It's provided also in your in the RFP itself, and there are specific email addresses for the different topic types. Or you're feel free to ask them now, which you have been. And I guess that moves us into answering your questions.
Great. Well, thank you so much, Jill and Pri and Lucas and Elliot, for walking us through this year's RFP. It definitely was an informative session, and we'll dig into the Q&A now to get people's questions answered.
So the first one is, do you plan to hold B2B meetings to connect researchers who are working on different aspects covered by the call? Jill, unfortunately this year we're not holding one of those calls. Is that correct?
Correct. We may be presenting some options for people to help identify, like some kind of networking sessions. Those have not been published yet, but we are working to try to put something together for people that are looking to identify collaborators.
So let me wrap up this last question really quickly and then I'll pass it back to you. But we also have a alternative protein research directory, and I highly recommend signing up for that and taking a look. Maybe that's a good area to find collaborators that are already signed up within our system.
Wonderful. Thanks for stepping in, Pri. I appreciate it. It looks like we have a question regarding, can a for-profit organization qualify as a new to the field researcher, and how is new defined? I think you may have addressed that question, but it's probably worth repeating.
And I think I typed it out also, but new to the field of alternative proteins is somebody, an organization that has not received any funding. I shouldn't say an organization. It should be an individual that has not received any funding for alternative protein work. So that could be funding that was a grant. It could also be industry funding. So they should be new to the field in that context. And we are, for collaborations and applications, open to all organization types. So
that's great. Thank you. And also, someone would like to know if there's a time restriction as to when the funded project can start, and can proposals be an add-on to an already funded project from other funders?
Yeah, so it would need to, a project should be able to start within 3 to 6 months of our funding, or our submission deadline. So that is the window that you should be available to start a project. Adding on to an additional or an existing project would need to still be a discrete proposal. If you are leveraging something like findings that you've already accomplished from another research project, that's completely acceptable, but this project would need to be its own discrete project.
Great, thanks for the clarification there. And someone's wondering how they can verify eligibility of their approach if they're still left unsure and wondering after this webinar.
Within the RFP, there are specific email addresses for each priority area. And so you could send a very specific question about eligibility to the appropriate email address.
Great, thank you. I think we're going to move into some topic-based questions because we are getting short on time, and I want to make sure that we have the ability to address a couple of questions, at least from topics A, B, and C. So Pri, we'll start with you. Someone is wondering if they can propose the use of side streams to produce vitamins under the funding priority A.
Yeah, so we had a few of these questions about vitamins and antimicrobials, or even like a fiber-rich side stream, something like that. I want to say officially for the record, these are all really, really valuable things to be working on. So I'm really happy that you guys are doing that work. Unfortunately, for this priority A, we are laser focused on food industry, high protein side streams, and forming plant protein ingredients from that. Part of that is that we can't fund everything under the sun. So even if we think that something's really valuable this time around, that's exactly what we're focused on. So yeah, thank you guys so much for doing that work though.
Well, thanks for that clarification. And is the call limited only to the crops that you mentioned, or can other waste crops also be used, assuming they have quick scale up potential?
Please, yes. So we got a few questions about, how do you define high volume? How do you define it as commercializable? How do you define it as a large industry? And honestly, these are meant to be a little bit vague because I want you guys to kind of convince us, give the case for your raw material. I understand that as a European American, I have not seen all the crops of the world that are being used. I don't understand that completely, and I don't want to, not be able to look at a side stream that I'm unfamiliar with, but regionally has a lot of importance, a lot of volume. So please make the case for it. We want to hear it. That being said, like, of course, if it's incredibly low volumes anywhere, then that's something to reconsider as far as applying or not. So yeah, I would say just like make the case as best as you can for your raw material, and we will definitely consider things outside of that side stream analysis report that I said everyone you should look at. There's only 8 crops that we evaluate in that, and we're definitely open to many, many more.
Excellent. Thanks, Pri. And I know there are additional topic A questions, but we're going to move on to priority B. It looks like there's a question here for you, Lucas. Could you get a definition, rather, of precision fermentation? For example, must the microorganisms have been genetically engineered?
Yes, that's a great question. There's also a question here. Is the funding strictly restricted to precision fermentation proteins? So let's start with that one. Yes, we are focused purely on precision fermentation proteins. So biomass ingredients would be considered out of scope. If you can show application to precision fermentation proteins, and maybe it can be related to biomass fermentation as well, that would be terrific, but the focus is on precision fermentation proteins. Regarding the genetic engineering, no, it does not need to be considered genetically engineered. The definition of precision fermentation is sometimes hotly debated. We're just considering any specific protein that is produced and concentrated or isolated from a microorganism to be a precision fermentation protein. We do ask, though, that the protein of interest be focused on improving the organoleptic properties of alternative meat products. And so we would also not consider food processing aids like enzymes to be within scope, because we want these ingredients to directly go to the product themselves. Excellent.
That was very helpful. And now we'll move to topic C. It looks like there are a couple of questions here for you, Elliot. Can fermentation-derived products or ingredients be considered under priority C?
Yeah, so if I'm interpreting this correctly, we're totally open to like microbes being the raw material that you would hydrolyze into, an amino acid source for cultivated meat media. I do want to say that, there are existing things like yeast extract or yeast lysate that, can be used. And I think, you run into we have to narrow the scope of this proposal to really focus in on hydrolysates. And so, and the production and processing, of hydrolysates. And so taking a simple lysate that doesn't really involve that process is sort of out of scope if that's going to be the primary purpose of your proposal. Now, certainly if you're using a microbial source like yeast and you want to compare like a yeast, a new method for yeast hydrolysis compared to like just a traditional yeast extract, that could be in scope. Like it has to incorporate these elements of the hydrolysis process and improving upon that. Hopefully that's somewhat clarifying.
Yes, thank you. And this final question, I'm not sure who the best person to answer would be, but it's a good one. Would it be possible to combine items A and C, for example, by using a food processing side stream as a source of amino acids?
I'll just say for priority A, the end product is a plant protein ingredient that can be directly consumed by humans. So for priority A, that wouldn't qualify.
Yeah, I think there's been a couple of questions around like, can we combine across priorities? I would really encourage people to focus on one priority. We have limited funding. You're going to be in a competitive pool. So please focus in on exactly what we've written in here, and you'll have the higher chances of being funded. So for instance, for that question, if you're talking about amino acids and the application is in cell culture, then sure, you can use a side stream as the starting raw material. But if the application is in cell culture, then it's priority C. It's not priority A.
Please look at the RFP that we launched, the actual document. There's an FAQ section there, so please look at there for your questions. And then there's also email addresses that are aligned with each of the priority areas that you can ask additional questions to.
All right, with that, we'll wrap up. Thanks, everyone. Bye-bye.